Welcome back. Now we already saw what mitosis was. That was an equational division. Now when you're talking about equational division, well, it was said so because in mitosis we had 2n as a parental cell and the daughter cell that was formed was also 2n. And that was a procedure, that was a process which was performed by the somatic and the stem cells. Now that is not the case wherein we are talking about meiosis. Well, meiosis is going to be very different from mitosis. Now when you talk about meiosis, well, meiosis itself is divided into two stages. We have stage 1 in which we have diploid cells. They will undergo reductional division. And when they undergo reductional division, it means that 2n will now form haploid. And these haploids will now undergo equational division to form 4 different haploid cells. Well, this reductional division is called as meiosis, and this equational division is also called as meiosis. But what is the difference? Well, this reductional will be called as meiosis 1 and this equational will be called as meiosis 2. Now you would be thinking that meiosis 2 is equational and equational is also called as mitosis. Yes, meiosis 2 is exactly similar to what mitosis is, and meiosis 1 is very different from mitosis. Well, that's wherein we'll understand. So we have these chromosomes in meiosis which will duplicate once, but the cell will divide twice. Instead of producing 2 daughter cells, we will have 4 haploid daughter cells. A diploid parent cell will undergo reductional division and equational division to form 4 haploid cells. And then these 4 haploid cells will be called as the gametes. This means that meiosis is performed by the egg cell and the sperm cell together called as the germ cells, which are now going to be the gametes of our body. So when we have these males and the females, well, they have diploid cells, that's 2n. When these diploid cells will undergo meiosis, that is the germ cells will undergo meiosis, they will form haploid eggs, that's haploid ovum, and they are going to form haploid sperm. Now when these haploids, egg and sperm, when they fuse together during the process of fertilization, they form a zygote which is diploid. So n plus n gives 2n, and this is the diploid zygote. That is why they are made as haploid, because we need to maintain the consistency of the chromosome number. We are 46. Let's say even the germ cell has 46. So 46 from the mother, 46 from the father, we will have 92 chromosomes. What are we going to do out of these 92 chromosomes? Well, we can't do anything. We'll become something else other than humans. And that is why, to maintain the species-specific number of chromosome, the germ cells which are involved in fertilization will then undergo meiosis. And by undergoing meiosis, they become haploid, so that when they come together, they form a zygote which is diploid. That is why this meiosis is highly essential. This meiosis will allow for sexual reproduction within the eukaryotic cells. That's why these ovum and the sperm need to be haploid, and they are the gametes which will make sure that the zygote is formed, which is of the same number. Now when you talk about meiosis 1, well, meiosis 1 is very different. Of course, it is going to have the normal stages of PMAT, that's prophase, metaphase, anaphase, telophase, but at the same time, it is going to be very different. So let's see. We have the meiosis 1 meiotic phase as karyokinesis 1 and cytokinesis 1. Now when you talk about karyokinesis, this is divided into the 4 stages of PMAT, that's prophase 1, metaphase 1, anaphase 1, telophase 1. The first stage is prophase 1, but the problem is that this prophase 1 is typically longer. It's going to be a very long cycle and it is going to be more complex. When you are comparing the prophase to the mitosis, it is going to be very, very different. This prophase itself is divided into 5 substages, and the 5 substages are leptotene, zygotene, we have pachytene, we have diplotene, and the end we have diakinesis. What is it? It is leptotene, zygotene, pachytene, diplotene, diakinesis. Again, leptotene, zygotene, pachytene, diplotene, diakinesis. So these are the 5 stages within the prophase 1. And because it is so long, that is why the meiosis prophase 1 is going to be typically longer and complex. So let's start with the prophase 1 of meiosis 1 and the first stage, that is leptotene. Now when you talk about leptotene, it can also be called as leptonema. And this leptonema, in which lepto means thin and nema means thread, in which you have the germ cells, of course they will have the chromatin network, and this chromatin network, just like the normal, will condense, and when they condense they form thin thread-like structures, and this, when forms thin thread-like structure, starts to look like the chromosome. Then we have the second phase of prophase 1, which is called as zygotene, also called as zygonema. Zygo means paired and nema means thread. So we have these 2 chromosome, thin thread-like chromosomes like here. Now they will look at each other, and then of course they will pair up as one. They will now come closer to each other, and when they come closer to each other, they have now become a homologous chromosome. Means same. Homologous means same. So they will become homologous pair of chromosome. Now still they are haploid. They need to undergo diploid stage because these are happening within the gametes. And of course these gametes are haploid, so they need to become diploid. So when they need to become diploid, that is they want to multiply, that's wherein they will undergo the other stage, and that is called as, let's see what it is. So in here we have the pairing, and this pairing is called as synapsis. It's not the synapse of the normal neuron, but it's very different. This is synapsis, and in this we mean pairing up the 2 homologous chromosomes. These chromatids over here are not visible. So we are not talking about sister chromatids here. We are just talking about the thin thread of chromosome. Let's move on to the third phase of prophase 1, which is pachytene. Now this pachytene can also be called as pachynema, which means thick thread. So something which was thin, now it has again multiplied, it has developed, and it has become a thick thread with the centromere. Do you see this? Now this is nothing but the sister chromatids. So they have developed into these sister chromatids, and these sister chromatids are now going to undergo another stage wherein they will combine with each other. They will cross over with each other. That is, one will come over the other. And that is going to happen in pachynema. Now just look at this. Do you see these sister chromatids over here? Well, they are not going to come over each other. It's not going to be one chromosome coming and crossing over each other. Rather, there is going to be a crossover between the homologous chromosome. See, these 2 are sister chromatids, and these 2, the orange ones, are sister chromatids. They are not going to cross over amongst each other. Rather, they are going to cross over on the homologous pairs. And that is why the stage of synapsis had to come. So the thin threads initially came together, they saw that they were homologous, and then they divided into sister chromatids. Rather, then they multiplied into sister chromatids, and that's wherein they'll form this, and this is called as the event of crossing over. Now, because this crossing over between the homologous chromosomes, and they are non-sister chromatids because the orange and the blue is sister chromatid. See where the crossing over is happening. It is in between the blue and the orange. They are all non-sister chromatids, but they are homologous chromosomes, and that's wherein this condition of chiasma or chiasmata is happening. Well, this event of crossing over is called as chiasmata or chiasma. Now, when you see here, when they have crossed over, of course, they will exchange the genes. They'll exchange some or the other kind of genetic information. And that's wherein the blue has small part of orange and orange has a small part of blue. Now, this exchange leads to recombination. And this recombination, which means the two chromosomes have recombined. Initially they were different. Now after crossing over they have become different. This leads to variation. Well, that is why we are not exactly like our parents. We are not exactly like our mother and father, but we have some variations. We have characters like them. We might be fair like them. We might be tall like them. But there will be certain characters which are not like them, which are ours completely. And why do they become ours completely? Well, they become ours only because of one reason. And that is because of this recombination. That is wherein the species have variation. That is wherein the diversity amongst us comes in picture. Now, let's move on to the next, that is a diplotein, also called as diplonema. Well, diplo means two and nema means thread. So after exchange, the recombined chromosomes will now start to separate each other. That is, they'll start repelling. And now because they have a variation in them, that is wherein the nuclear membrane and the nucleolus will start disappearing. So when the nuclear membrane and the nucleolus start disappearing, that is the moment they will enter into the last stage of prophase 1, and that is diakinesis. And in this diakinesis, the entire thing is going to move. That is, there are going to be chromosomes which are now going to condense, and that's wherein they will start moving. That's wherein the nuclear membrane will disappear, nucleolus will disappear, and that's wherein the entire prophase 1 will come to an end. It is a terminalization phase, and that's when everything comes to an end. The spindle fibers will start to appear, and once the spindle fibers start to appear, then we know we enter into the metaphase. So we have the metaphase 1, in which the recombined chromosomes will now appear as a single thread. That is, they'll appear in the single line. That's the equatorial plane, and that's wherein they will be pulled apart. Now this is the equatorial plane. They are getting organized along the equatorial plane, and of course the spindle fibers will go and attach to the centromere. So when they attach to the centromere, this is going to be similar to the mitosis, and then they enter into anaphase 1, wherein they are pulled apart. Spindle fibers will pull them apart, and of course the homologous chromosomes will go to the two opposite poles. Along with that, there is this procedure called as a disjunction, and this disjunction will make sure that the half, that is the end half of the number of chromosomes, will gather at the respective poles, and that's wherein another nuclear membrane will develop. And when this nuclear membrane starts to develop, that's wherein we understand that yes, now is the time for telophase. So this nuclear membrane has developed, spindle fibers will disappear, and of course these homologous chromosomes with recombined part of the chromosomes will start to decoil, and when they decoil or when they uncoil, again they'll form these chromatin network. Well, we have the nucleolus, the Golgi, and the endoplasmic reticulum, which are being formed because they are a part of these cells. Nuclear membrane will start to reappear. Well, that was the last part of karyokinesis 1. And now is the time for cytokinesis 1. So after all this, there are two haploid daughter cells with single set of chromosomes which are going to be formed. That's cytokinesis 1. Now, cytokinesis 1, the moment it ends, then meiosis 2 has to start. But a very short resting phase between meiosis 1 and meiosis 2 is going to be there. That's going to be a very small gap, and that gap is called as interkinesis. And the moment interkinesis ends, we have the start of meiosis 2, which is exactly similar to mitosis. So that's a revision for mitosis. They are divided into 4 stages: prophase 2, metaphase 2, anaphase 2, and telophase 2. And in all this, we have both haploid daughter cells which will undergo meiosis 2, and then they will form 4 haploid germ cells. Now let's see how that happens. We have prophase 2, in which every chromosome is again, they are going to form these condensed structures, and again when they form these condensed structures, they will get converted into the chromosomes. Now when they get converted into the chromosomes, this is how they'll appear. They'll start appearing with their sister chromatids, though not very fair, but they will still appear. The nuclear membrane will start to disappear, the nucleolus will start to disappear, the centrioles will duplicate, and of course they will move towards the end of the poles. The nuclear membrane will start to disappear, though it will become very light, it will not going to be disappear very soon, and the nucleolus is the same. It starts to disappear. That's wherein we know it's the end of prophase, and that's wherein we will enter to metaphase. Now in the metaphase, all of these recombined chromosomes will now make sure that the nucleolus and the nuclear membrane are completely disappearing, and then that is the time these equatorial planes will start to form. The chromosomes will become completely visible, and they will all be arranged in a parallel line that is along the equatorial plane, that's the central plane of the cell. Now the spindle fibers will be formed from the centriole, and they will go and get attached to the centromeres, and that's wherein they go and attach to the centromeres and they pull them apart. The centromeres will split them apart. When the centromeres will split them apart, of course they'll go towards the end, and when they are going towards the end, they are called as the daughter chromosomes. Now these daughter chromosomes that have reached the poles, they will start developing a nuclear membrane, and that's what they appear like. They again appear like a bunch of bananas. Now these chromosomes which have reached the opposite poles, again they start to decondense, and then another kind of the nuclear membrane is going to appear. The nucleolus is again going to appear, and that's wherein the telophase ends. Cytokinesis is completed. Spindle fibers completely disappear. Karyokinesis is completed, and cytokinesis begins. Now when cytokinesis begins, again there is going to be a notch. This notch is going to deepen, two cells are going to be formed, and these two cells are called as the daughter cells, which will again go further and divide more into 4 cells. Well, this is how cytokinesis 2 ends, and meiosis 2 with the 4 germ cells, and all of them are haploid, will be present. Well, that is how meiosis 2 occurs. Meiosis 2 is exactly similar to mitosis. There's no difference as such. The only difference between meiosis and mitosis is in meiosis 1, wherein there is genetic overlapping, and this genetic overlapping is merely because of one reason, and that is because we need variations. Well, that's how the germ cells will be formed.